Hi, I'm Ann Cooper. I'm a student at the University of Kentucky. I really enjoyed the idea of this class because in the past I had the opportunity to present a project at Kazan, Russia, and it was very exciting to meet someone outside of the U.S. I also had the opportunity to work with Professor Dr. Kim at Chongnam National University in South Korea, and he's actually helping me with the project that I'm going to be presenting today. So I hope you guys enjoy the project, and I would love any feedback that you guys have. Alright, let's go to the presentation. So I'm looking at the effects of enhanced sensory perception on learning and memory retention in Drosophila. So there's two types of learning. There's explicit learning, which is the conscious recall about past events. It's well developed in the vertebrate brain. And implicit learning is memory for motor skills, and this is without the conscious recall of past experiences. So why study the Drosophila? There are many similar genes between Drosophila and mammals, and to learn more about learning, we'll have to study simpler animals such as the Drosophila, and in my project, I'll be looking at the larval stages. So here's the life cycle of a Drosophila. We have the embryo, the first instar, second instar, third instar, prepupa, pupa, and then the adult. So first instars, second instars, and early third instars tend to stay in the dark, so deep in the food, and then once they become late third instars, they climb out of the food and then pupate outside and then become an adult. So what my first question is do the larval stages show abilities to learn? So the first, second, and early third instar larvae. Studies have shown that the adult Drosophila do show abilities to learn, but how about the larval stages? So here's my learning setup. I have a plain agar petri dish on this banded pattern of light dark. So the early instar larvae tend to prefer the dark, and I'm trying to teach them to go towards the light. So I have another petri dish in the light that contained fructose, which is a food that they like to go to. And then in the dark, I would have quinine hemisulfate, which is kind of a salt that they try to avoid. And I would just have this box with the felt material and then just close the box when they're supposed to be in the dark. So here's just a little diagram of my experiment. I have the preconditioning phase when they're in this agar for 2 minutes. And then I would mark where the larvae was every 30 seconds. So if they were in the light or if they were in the dark. Then I would transfer them to this other petri dish that contained fructose for 2 minutes, let them crawl around, and then transfer them to the quinine hemisulfate in the dark, let them crawl around for 2 minutes. I'd switch the larvae back and forth, and then after that I'd put them on a new petri dish on this banded pattern of light dark for 2 minutes and record it every 30 seconds if they were in the light or if they were in the dark. So here's my graph for the first instar larvae. These are the preference values and the learning index. And what these graphs show, so the positive is the fructose in the light, and then the negative is quinine hemisulfate in the dark. And then I also switched the foods, so I had quinine hemisulfate in the light and then fructose in the dark. So these are two groups of the Drosophila larvae, first instars. So these positive values indicate they like the light, and the negative values indicate they preferred the dark. So you can see here the dots here are the mean, so even the first instars preferred the dark as well as for when I switched the food for quinine sulfate in the light and fructose in the dark. For the learning index, if the mean is above 0, it indicates that the larvae did show abilities to learn. However, this is not significant, so my first instars did not show abilities to learn. So moving on to my second instar, again here are the preference values. This was significant at 0.05. They preferred the dark, and then here as well they were at 0, so they didn't really prefer anything. The mean value for this learning index was below 0, so did not show abilities to learn. So I conclude that my second instars did not show abilities to learn. For my third instar larvae, again here are the preference values. Looking at the learning index, the mean is above 0, however this is not significant. So my third instar larvae did not show abilities to learn. So then I began thinking that maybe quinine hemisulfate is not a strong negative food for the larvae, and maybe I should have a stronger negative reinforcement. So Dr. Kim in South Korea has these genetically altered Drosophila that are capsaicin sensitive. With this, capsaicin is the active ingredient in red peppers. It works on the same receptors as heat, so that's when you eat spicy food, you feel hot too. So I looked at capsaicin sensitive larvae and compared it to my background larvae. So just to show you how sensitive the larvae are to capsaicin, this first one is the capsaicin sensitive larvae. You can see this rolling effect, whereas the control is not really affected at all. The same rolling effect can be seen when you place a hot probe towards the larvae and it would roll away from this hot probe. So the capsaicin sensitive larvae are very sensitive to this capsaicin. So my second question is, do the capsaicin sensitive larvae show abilities to learn throughout development with one-time learning? So here's my previous diagram for my first question, and then for my second question. The things that are different is instead of quinine hemisulfate, I replace this with a high concentration of capsaicin, and instead of switching them back and forth 5 times, I switch them back and forth 1 time. So here is my graph for the first instar larvae. These are my background preferences, my capsaicin sensitive larvae preferences, and then the learning index. So mainly just focusing on the learning index. Both the background larvae and the capsaicin sensitive larvae, the mean was at 0, indicating no learning. So my first instar larvae for the background and my first instar larvae for the capsaicin sensitive instars did not show abilities to learn. Looking at my second instar, mainly focusing on the learning index, my background larvae did show abilities to learn, however not significant, so my second instars did not show abilities to learn. Capsaicin sensitive larvae, the mean is at 0, so they also did not show abilities to learn. For my third instar larvae, the background larvae, the mean was at 0, and then the capsaicin sensitive larvae were below 0, so both did not show abilities to learn. So my third question is, maybe the larvae it's too much of a short period of time to test if they do show learning, and maybe they retain the memory and then express it later on. So what my third question is, do the capsaicin sensitive larvae show memory throughout development? So I would train them as a first instar, wait until they became a second instar, and then did the post-conditioning phase when they were second instars. I also did first instar to third instar, and then second instar to third instar. So here are my graphs for when I trained them as first instar and then tested them as second instar. So these are just the preference values for the first instar for both background and capsaicin sensitive larvae, and then these are the second instars for my background and capsaicin sensitive larvae. My learning index for the first instar larvae did not show abilities to learn. Looking at when they became a second instars, there was a significant difference between my background larvae and the capsaicin sensitive larvae. So my background larvae did show memory retention, and then my capsaicin sensitive larvae did not show abilities for memory. For my first to third instar larvae, so trained them as a first instar and then tested them as a third instar larvae. For my first instars, my background as well as the capsaicin sensitive larvae were either at 0 or below, so did not show abilities for learning and memory retention. And then for my third instar larvae, the background was above 0, however not significant, and my capsaicin sensitive larvae stayed at 0. So both background and capsaicin sensitive larvae did not show memory. And then for my second to third instar larvae, mainly looking at the learning index, when they were tested as second instar larvae, background was above 0, however not significant, capsaicin sensitive at 0, And then when I tested them as third instars, both background and capsaicin sensitive larvae were either at 0 or below. So my background did not show memory, my capsaicin sensitive did not show memory. So in summary, for the larval stages did not show abilities to learn. This may be due to remodeling of the brain since it grows extensively throughout development. So here's first instar larvae brain, second instar, third instar. So you can see the significant growth of the brain. So there may be a lot of remodeling going on. So maybe larvae do not show abilities to learn through all these, through the development. For my background in capsaicin sensitive larvae, the background larvae did not show learning through development as well as my capsaicin sensitive larvae. For memory, from first to second instars, the first instars in both the background and capsaicin sensitive larvae did not display associative learning. The second instars in background stain did display associative learning, therefore memory. However, the second instars for the capsaicin sensitive larvae did not display memory. For memory from first to third, background and capsaicin sensitive larvae did not display memory. And then memory from the second to third instar larvae, both did not show memory. So some notes to consider. Since the significant tests were non-parametric, a sample size of 10 is considered small to gain any significance. The variance of data for the preference values may have influenced the learning index. So there's a large variety, and then here are the mean values, little x. So due to this high variability, it could have influenced my results. And then also, this was adapted from a previous paper, Gerber et al. In 2004, that did show that larvae had learning abilities, and then I changed their setup of this pie chart to this striped area because I thought if the larvae would just stay in the dark, they would never experience the light, whereas here the larvae can crawl and then see the light on either side. And then a student from Cold Spring Harbor that came to visit the lab said that it was very hard to reproduce the same data. So mainly looking at their learning index, they did show there were learning abilities, and then mine was not significant, so I couldn't conclude that there was a learning index. Some of my future goals: I want to increase sample size so I could possibly gain significance through the significant testing I use, increase the time spent on either the positive or the negative stimuli for the capsaicin sensitive larvae, vary the concentrations of capsaicin. Maybe the concentration I used was too strong, so that's why the capsaicin sensitive larvae didn't show an influence of the negative stimuli. Test at different durations of conditioning, so I had about 2 minutes on either the positive or the negative stimuli, so maybe changing the time frame may show learning in larvae. Conduct similar research for other genetically mutated fly strains. Repeat parts of my studies with temperature conditioning instead of the taste, and see maybe if the temperature aversion can be overridden with food attraction. So I just wanted to thank the Kentucky Human Researchers Program for helping me fund the project, Dr. Kim for the genetically altered Drosophila and helping me understand the genetics behind it, and then the lab at the University of Kentucky. So thank you for watching, and I look forward to your projects.